Yeah, thank you, Sarah and Aviv, for a wonderful tour de force through only 3 years of history in the Human Cell Atlas. And it's amazing to see how much has happened since the first meeting in London only 3 years ago, and much more to happen in the next few years and decades to come. And towards that, the first session of this Human Cell Atlas meeting will be about building the HCA Roadmap, and we will have 4 fascinating presentations shedding light on different aspects of the Human Cell Atlas Roadmap. And the first one to kick off is from Monique von der Wijst, who will discuss and share with us the ideas about sample selection in the Human Cell Atlas.
Good afternoon, everyone. First of all, I would like to thank the organizers of this meeting for giving me the opportunity to talk here. So I'm Monique van der Wijst, and I work in the Department of Genetics in the University Medical Hospital in Groningen in the Netherlands. And we are very much interested in how we can use single-cell RNA sequencing data for a personalized disease understanding. So in our lab, we are interested in linking genetic variation to disease risk. And in the past decade, many scientists have been performing genome-wide association studies, which led to the identification of over 10,000 of genetic risk factors that can be associated with an increased disease risk. But the main issue with these GWAS studies is that they do not really tell you how these genetic risk factors are associated with disease. This very much remains a black box. So we believe that population-based cohorts can be very valuable in order to uncover this black box. And they can help us understand which genes and which pathways and in which specific context genetic variation can lead to disease. So within our institute, we have initiated such a cohort called Lifelines. And in this cohort, we have now 167,000 participants from the 3 northern provinces of the Netherlands. So together, these individuals make up around 10% of the entire population of these 3 northern provinces. So within this cohort, we have 3 different generations, and we keep following people for 30 years over time. And every 5 years, we are collecting sample material, and we're measuring, for example, BMI, and we are collecting questionnaires. So the main advantage of this cohort is that it's a recallable cohort. So if we are interested in a particular research question, we can just select a subset of this cohort and invite them to collect additional research material. So within Lifelines, we also have another subset of individuals containing 1,500 individuals that are in the Lifelines Deep cohort. And this cohort, in this cohort, what we have is that we have genotype information, we measure methylation levels, gene expression levels, we collected stool samples and determined microbiome composition. And this now really has allowed us in the past to successfully link genetic variation to, for example, methylation levels and gene expression levels. But what we really learned from these studies is that the link between genetic variation and, for example, gene expression is very much context and cell type dependent. So that's why a few years ago, we decided to also start generating single-cell data within this Lifelines Deep cohort. So right now, we have single-cell RNA sequencing data of 165 individuals within a subset of this Lifelines Deep cohort. What we did is that we collected blood material and isolated PBMCs. And these PBMCs were either kept unstimulated or stimulated with 3 different pathogens for 2 different time points. And for each individual, we selected around 1,000 cells for single-cell RNA sequencing. So right now, we have around 1.1 million single cells available. And what we would like to do with this data is that we would like to determine the link between genetic variation and gene expression levels and the gene regulation. So for this, what we're doing is that we're performing EQTL analysis in the single cell data, and we are reconstructing personalized gene regulatory networks. So when you do an EQTL analysis, what you're looking for is that you're looking for genetic variants that are modulating downstream gene expression. And if this downstream gene is located within a 1 MB window from the genetic variant, we call it a cis-EQTL, whereas if it's further apart or even on a different chromosome, then we call it a trans-EQTL. So we now know that many of these EQTL effects are cell type and context dependent. And that's why it makes a lot of sense to start doing single cell or start using single cell expression data to unbiasedly identify these kind of EQTLs. So over here I show you an example. So last year we published a small pilot study in which we collected blood from 45 individuals. And we could show proof of concept that EQTL analysis is feasible in single cell expression data. So I just give you one example shown over here. What you can see is the expression of a gene called T-span13 on the y-axis. And you see 3 different genotypes individuals can have at this specific position in the genome. And what we show over here is that only in the CD4 T cells we see that this EQTL effect is present. So people with the AA genotype tend to have a lower expression of this gene compared to people with the TT genotype. What is important to mention here is that if we would have analyzed this single cell expression data as if it were bulk expression data, so averaging all the cells together, then we would not have picked up this EQTL effect. So really showing that the single cell expression data adds a lot of value here. What we did as well in the study is that we could discover that genetic variation not only affects downstream gene expression levels, but it can also affect how 2 genes are being co-expressed with each other. So what we learned from these studies is that yes, we need many different individuals in order to do this kind of analysis, but we cannot just do this by ourselves. So that's why we set up the single cell EQTLJ consortium. So in this consortium, we are mainly focusing on blood cells. And the reason for that is that right now, most of the population-based single cell studies have been performed in blood. So it's a nice test case to start with. And what we would like to do is identify EQTLs in the single cell data. So either cis-EQTLs, trans-EQTLs, or response EQTLs. In addition to that, what we also would like to do is reconstruct personalized gene regulatory networks. So right now we have 21 different groups that are participating in this effort. And together we already have the availability of over 5 million single cells from more than 1,600 different donors. But the main issue here is that we're working with privacy-sensitive information. So we are working with genotype information from all these individuals. And you cannot just share this information across cohorts. So what we are doing instead is that we're bringing the algorithm to the data. So this allows each cohort to analyze their data by themselves, and then we can just share the summary statistic data and analyze this in a centralized place. So we believe that the outcome of this consortium can really help in the clinics in the future. So first of all, we expect that we can identify those cell types that are associated with particular diseases. And by reconstructing these gene regulatory networks, we can start to identify those genes that are really driving a particular disease. And by then implementing this personalized or genetic information, we can start predicting who's going to respond to a particular drug and who's not. So with this, I would like to summarize my talk, and thank you for listening. So what I discussed is that deeply phenotyped population-based cohorts are required to gain a personalized disease understanding. And we are now going to gain a deeper understanding in how genetic variation is affecting gene expression and gene regulation through our initiative, which is the single-cell EQTLJ consortium. And because we're working with this privacy-sensitive information, we are bringing the algorithm to the data, so this allows worldwide collaborations. And we believe that the acquired knowledge will be valuable to move from more traditional to precision medicine in the future. So with this, I would like to thank the people in the lab, all the participating groups in the single-cell EQTL consortium and BBMRI, and all the individuals that participated in our Lifelines cohort. And I would like to end this talk by saying that if you're interested in joining our efforts, then please get in touch either with me or via email or Twitter, and you can always visit our website for more information. Thank you.
Yeah, thank you, Monique. A wonderful link between human genetics and single-cell community.
Yes.
So are there questions to Monique? Yeah.
Thank you. Just a curiosity, in your cohort, do you also have availability of any biological samples, clinical data?
Can you repeat the question? It was hard to understand.
Can you hear me now?
Yes.
Okay. I was wondering whether you also have access to biological samples from this cohort, clinical data eventually related to disease states, or if it's just availability of genetic information that you have access to.
So in the Lifelines cohort, people fill in questionnaires, and there they also state if they have any diseases or just got some diseases. And we collect blood material and urine samples, I believe. And well, we are able to re-invite those individuals. So if we would be interested in a particular sample, it might be possible. Of course, we cannot just take lungs or something, but I mean, blood or urine samples, that is available.
Okay,
Yeah.
Thank you.
So what about, I mean, because it's a single-cell EQTL, so what about in a single-cell MEQTL extension of your consortium? So adding single-cell ADAC-seq, for example, or
Yes,
single-cell whole genome bisulfite sequencing to this. So you could repeat this, in particular, if you're interested in the environmental induced changes and impact on gene expression at the single-cell level.
Yes, so we are now also thinking of not just collecting gene expression data, but also starting to collect different data layers. And ATAC-seq data is one of the layers that we are currently also planning to start generating. And also, in order to reconstruct these gene regulatory networks, it will be important to have different layers of information available. So ATAC-seq data is certainly one of them. Yeah.
Okay. Yeah, there's another question in the back.
Yeah, I had two questions. One was, what are the stimuli that you're using in vitro? And the other was, you used kind of bulk CD8 cells to analyze, or bulk CD4s. Can you split those up in any way, or does that give you any additional information?
So the first question was, let's see.
What kind of stimulation?
Oh yeah, what kind of stimulations we used in our data sets. So the data set I showed you, we stimulated with Candida albicans, which is a fungus, and Mycobacterium tuberculosis, and Pseudomonas aeruginosa. And the second question was
whether you could get more resolution on the different types of T cell or other types of
yes,
cell.
so right now we did this EQTL analysis by just pooling all the CD4 T cells from one individual together and taking the average gene expression. But what we thinking about where we want to move to is get more gradients, so not just say this is CD4 T cells, but for example incorporate pseudotime information, so when we start stimulating cells, get some kind of pseudotime over this response, and then get more resolution and really pinpointing when particular EQTL effects are present.
Thanks.
Yeah.
Okay, so thank you very much again, Monique. And I think in the interest of time,